This video will cover a variety of topics regarding biopharmaceutical properties. Those are properties of drug substances that relate to their ability to work effectively as drugs. So we talked about in functional group analysis many features in pharmaceutical properties such as solubility, log P, and ionization. And each one of these things comes about as a natural consequence of the functional groups that are present in a molecule and the relative relationship to one another. So one of the things that's important with regard to this is this can control a whole variety of materials such as the stability, the ability to formulate a drug in a particular way, and a possibility of being able to administer a drug in a particular fashion. Years ago, one of the biggest problems that we had in drug development had to do with things failing in commercial development based on poor pharmacokinetics and bioavailability. So what has happened since then is the pharmaceutical industry is focused on predicting these features ahead of time so that compounds which go into development do not have features which are incompatible with effective drugs. And as you can see in this short period of time from 1991 to 2000, they were able to decrease that as a reason for why compounds failed when they went into clinical trials. Obviously, since this time, the pharmaceutical industry has focused on some of the other issues that are larger issues with regard to things making it into development, and they've made progress on that as well. But we're focused here on these biopharmaceutical properties. So you can see how they very much got a handle on it when they recognized that it was an important aspect and could save them a lot of money not taking compounds forward that would not make it through. So we're going to start out just talking about simple things like water solubility. Water is a very polar solvent. Here's just a list of other solvents in comparison. And with regard to a polarity index, we can see water has a value of 10 versus hexane, which is 0.1. Hexane, of course, is a alkane. Toluene is an aromatic hydrocarbon. Methylene chloride is a polar, it's essentially methane with two chlorines in it, and chlorine is more electronegative than carbon, so it has a dipolar nature. And methanol is, of course, it's a methane with one of the hydrogens replaced by a hydroxyl group. So water has this partial minus charge on the oxygen and partial positive charge on the hydrogens, and that gives it a lot of unique features. Okay, one of these is that it's not completely it's tetrahedral in a sense, but the bond angles are distorted from a perfect tetrahedron because obviously it's not completely symmetric. So when we have a non-symmetric attachment to a central tetrahedral center, the arrangement of the bonds around it are not going to be completely perfectly tetrahedral. Now the other thing is it's involved strongly in hydrogen bonding. Here we show water molecules with the covalent bonds as solid lines and the hydrogen bonds as dashed lines. So when we talked about molecular interactions, what hydrogen bonds are. So these are hydrogens that are covalently attached to oxygens or nitrogens that are interacting with a lone pair of electrons of an oxygen or a nitrogen. So you can see in just bulk water, we can have a lot of strong interactions with all the other water molecules. They're able to diffuse and move around among one another, but it's not the same situation as you would have just in a hydrocarbon, right? Because there is some affinity for each of these waters to each other. The water can serve as an acid or a base. In the form of an acid, we normally talk of it as the protonated form of water, which we call the hydronium ion. So it's OH3 plus. So protonated water is that, and of course then it acts as a conjugate acid. Now, in the case of just plain water, of course, it can act as a base to pick up a proton, right? And then the result of that is the creation of a hydronium ion. It is a good solvent for many other functional groups. We're showing a methanol molecule here. So methanol has a hydroxyl group in it, and so it's a hydrogen bond donor and acceptor. What we mean by donor is that it actually has the oxygen that is covalently attached to the heteroatom, and that proton then can be donated in a sense, or let's say borrowed by another lone pair of electrons on another molecule. Whereas an acceptor is a compound that has a lone pair that can accept that partial interaction with the hydrogen that's attached to another heteroatom. Okay, so when we dissolve a hydrophobic compound, they're not involved in hydrogen bonds. What you have is you have a dipole, induced dipole interaction, right? The dipole is the water and the induced dipole is the hydrocarbon. Now, in that situation, as we talked about the hydrophobic effect, in that it tries to minimize its contact surface with water, and that is an entropy-driven process because the water that's arranged around the hydrocarbon is organized, and when there's less of the water that is in that state, in other words, when we have the hydrocarbon coalescing, it takes less water to encapsulate that hydrocarbon. So that leads to a net increase in the amount of water molecules that are essentially free in solution as opposed to being in that kind of cage around the hydrocarbon. So this is a very important principle we find in a variety of things, but protein folding is one important place where it comes into effect. Now, why do we care about water solubility? Well, our bodies are essentially water. So most drugs contain both hydrophilic and hydrophobic functional groups. And how soluble they are in water depends on a variety of things. It depends if those groups are charged. It depends if they're polar or not polar. Now, the relative proportion of groups that like interacting with water versus those which don't is going to basically give you the overall solubility of that molecule. Now, if a drug can contain an ionizable group, in other words, one that creates either a cation or an anion, then the water solubility is generally dependent on the ionization of that molecule. In other words, it's more soluble when it's ionized, and when it's not ionized, it may be more less soluble, basically. In other words, it can interact via hydrogen bonds and things like this potentially, but it cannot be involved in ion dipole interactions, for example. So the pH can be critical with regard to solubility. And when we're going to talk about how we estimate solubility, so we'll be bringing this feature up as we go through all the other topics. When we talk about solubility, we'll usually talk about solubility in terms of, oh, it's soluble, it's insoluble, it's very soluble, right? And so the question is, what does this actually mean in quantitative terms? So this table just roughly shows kind of a rough quantification of some of these terms. The only one we really want you to know on here is soluble at the 3% level, which would be like 30 milligrams per milliliter. Would be a value that would be associated with that. If you think about it, that's pretty reasonable because what if you take, say, a pill and say it has a tablet, and what would it contain? It would be something that would be anywhere from, say, 3 to 300 milligrams, right? So that means that as long as you have more than 30 milliliters, right, which is basically like an ounce of water, that it should be able to dissolve in that. So you know, that's a reasonable value. And normally you're having like a glass of water, which is several ounces. So your drug should be able to fully dissolve in that if it is deemed as a soluble material. USP specifically, so this is US Pharmacopoeia, specifically designs water solubility is greater than 3.3%, so 33 milligrams per milliliter. And there are some other definitions, but this is the one that we'll primarily use. Now, another thing that we're going to talk about quite a bit is what we call the octanol water partition coefficient. So P is actually the ratio between the amount of the concentration of a drug that dissolves in octanol versus how much of it will dissolve in water when the two are mixed. And normally we represent this value, since it can vary quite a bit in large amounts, we take the log of that ratio. So we talk about log P. Okay. And if we determine empirically, in other words, we do an experiment, we actually put a compound into a combination of octanol water, shake it up, let them separate it, and then actually measure how much is in each of those two layers. We call that a measured log P. You'll seldom see this value. You'll seldom see it reported like this, but this is just to let you know there is such a thing as you actually do an experiment to determine the log P, or you can just calculate it based on its structure. So in many cases now there are programs, and there are quite a few of these, that you actually put in a code for a structure, and it analyzes that and actually uses an algorithm to determine what the predicted log P would be. So we call that a calculated log P, or a C log P. You'll sometimes see the C log Ps. Most of the time when you see log Ps, it kind of depends what the source is. Now, most of the time it's going to be a calculated log P that you see, because that's just the easiest for people to do. The only place that a measured log P is usually going to show up is in the actual work that's done by, say, a pharmaceutical company to get approval for a drug. So they put together a packet of analytical information on the drug, and it has to be real data, and it has to be very complete. And so actually measuring the log P would be one of the things that would be reported in that packet. And so yes, that could be found in the information of a drug approval and then reported, but for that, just practically speaking, most of the time C log Ps are what people use. They'll see the structure and they'll just calculate it and then say, oh, that's what it is. Okay, so let's get back to just what is the log P and how do we use it. So the log P again is log of the concentration of the solute in octanol divided by the concentration of the amount of solute that dissolves in water. It's a ratio. It's dimensionless. It does not tell you how much of the solute goes in. In other words, it doesn't tell you about the solubility. What it tells you about is just the relative solubility of the two. Okay? So if you have a very soluble compound, right, the two concentrations could be large numbers. If you have a relatively insoluble compound, the two numbers could be low, but the point is you're still going to generate a dimensionless ratio, and so you're not going to know how much of that solute is in solution if you just see a log P. So a log P does not tell you solubility. And the reason why I bring this up is this very next line, in that there are guides and things that will tell you that if you have a log P that's less than positive 0.5, it is considered soluble. Okay? Take that with a grain of salt. It's generally true, but it's not necessarily true. Okay? So, what does that mean with regard to this ratio? This ratio is what? You would have a value of 0 if you have an equal concentration in octanol and water. A value of plus 1, right, which is greater than this 0.5, would be a value where your solute concentration in octanol is 10 times that of the solute concentration in water, right? Because your value would be 10, or 10 to the 1, and if you take the log of that, it's 1. So that's how you calculate these. So a value of 0.5, I haven't pre-calculated this, so it's just some number that's greater than 1 and less than 10, somewhere in there. that gives you, when you take its log, right, 0.5. And that's what's considered soluble. So again, it's just this relative solubility between octanol and water, and doesn't tell you the absolute solubility. Okay, so we have something called functional group analysis, and it's a means of potentially predicting water soluble. It's an estimation. It's not meant to be exact. There are two methods, an analytic method, which I'm not going to show you because it gives values, and again, it's just an estimation, but part of the problem with that method is all of the values that are needed to calculate a new drug that you might find are either very hard to find or haven't been determined. It uses values from a table where all the functional groups have not been fully characterized. So it doesn't make it as generally useful as this empiric method. So we're really only going to talk about this empiric method. And what it amounts to is it talks about solubilizing potential. And what that means is certain functional groups, and now this method identifies functional groups in a slightly different way in the way that we described it initially. So you have to take it into account that sometimes a particular method calls functional groups different things. Just like when we talk, well, later on we're going to talk about something called the Lipinski rules. It also identifies functional groups in a different way. We're not going to worry about that at the moment. So when you have a method like this, just follow its instructions basically. Okay, don't try to analyze it with regard to, oh, wait a minute, you know, we call this functional group something else in another place. Yeah, don't worry about that, only because this method is based on its own set of rules, so you just have to follow its rules. So what we talk about with solubilizing potentials is this: it's a balance between polar and nonpolar groups within a molecule. And if you essentially have more polar groups, they're going to interact with water better, right, than the nonpolar groups. And if you have a nonpolar group in a molecule, it's going to take a certain amount of polarity added to that in order to solubilize it. And that's going to be proportional to, the way we figure this is the number of carbons in the molecule. If we start out with the number of carbons in the molecule, which we can get from the molecular formula, we're going to have to say we're going to need a certain amount of solubilizing potential added to that in order to get it into solution. So the solubilizing potential is, we get that from numbers that are associated with the types of functional groups that are in the molecule. And we can simply add them up. Okay? Now, there's a table of these values, and actually there's two of them. There's one, if you have a small molecule and it has only like one functional group in it, that would be a monofunctional molecule. And so there's a table of values for that. We're not going to use those because most drugs, virtually all the drugs that we're going to be talking about and using, are polyfunctional. So there's a separate set of values which are slightly smaller. In other words, the effect of the solubilizing potential of it in a polyfunctional molecule is a little proportionally less than it is in a small molecule where there's only one functional group. So we're going to use the table of solubilizing potential that is applicable to polyfunctional molecules. Now, there's other things that can happen. Is you can have intermolecular bonds, right? And those are bonds that happen between molecules, and those have to be broken by water in order to solubilize a material, right? So if two molecules are bonded to one another, they have to come apart, and then water must surround each of those molecules separately. And then we have intramolecular bonds. Now these are between two different parts of the same molecule. So it's not necessary for them to be broken apart in order to solubilize the molecule. And so what is the significance of that? If we have intramolecular hydrogen bonds, the influence of those intramolecularly bonded polar materials might be a bit less than we would expect than if they were interacting with water. In other words, they don't need to interact with water because they're interacting with themselves. Now, in this empiric method, the empiric method really doesn't take this into account, but it is something we can recognize when we're like on a borderline situation. Okay, so we're going to get values here, and sometimes this value is going to say, it's either going to say it's soluble or it's insoluble, or it's going to be right near the dividing line, right? If it's right near the dividing line, then you can say, well, it may or may not be soluble, right? And some of these features like, oh, are some of the polar groups that it has involved in intramolecular bonds, that's going to push it towards the less soluble side. And you might say, well, if it's borderline, we might expect it to actually be insoluble in that case. So let's look at some of the actual calculations and what they look like. So here's estriol. It consists of a phenol and 2 alcohols. And this is what we're talking about with the solubilizing potential of using monofunctional group solubilization factors. So what this means is if we have a phenol, which is a polar group, right, it can solubilize the equivalent of 6 to 7 carbons. So what does that mean? Well, phenol itself contains 6 carbons, right? So it's only able to solubilize maybe itself and 1 more carbon. So its potential is, moderate. For an alcohol, it can separately solubilize 5 to 6 carbons. Okay, and we have 2 of them. So now instead of 5 to 6, it's a range of 10 to 12. So if we then add up the 6 to 7 plus 10 to 12, it predicts that you can solubilize 16 to 19 carbons. Now estriol has 18 carbons. So that actually falls within this range. And based on this, you might say, okay, it's possible that this material might have some solubility, but it's kind of marginal if you think about it. Okay? Now, notice those 2 alcohols. They're right next to one another. And if you notice, if we count from one of the hydrogens, so HO, and then we have 2 carbons in between them, and then we have another oxygen. If you imagine that there's a hydrogen bond between one of those hydroxyl groups and the oxygen of the other hydroxyl group, that makes a 5-membered ring. And remember, 5 and 6-membered rings are favored types of ring sizes. So that's actually a hydrogen bond which is likely to happen, and it is going to be intramolecular, which means that's going to be less available to forming another hydrogen bond with water. So in a sense, this is one of those situations where we might say, well, our solubilizing potential is marginal, and we have this intramolecular hydrogen bond. Maybe it's going to be not so soluble, right? We might predict it not to be soluble, even with this monofunctional, right? The monofunctional makes it a little gray area. Now, if we take into account that, well, this is really a polyfunctional molecule, why don't we use the solubilization potential for polyfunctional molecules, right? And let's see what kind of a value that gives. Now, this is what we're talking about, this intramolecular hydrogen bonding. Okay. And there's hydrogen bonds here, here, and here, but because of that intramolecular hydrogen bond, notice there would be other potential hydrogen bonds that that OH could be involved in that it wouldn't necessarily have to be because it's interacting with itself. Okay, so let's look at the polyfunctional. Phenol is 3 to 4. It's less, isn't it? And alcohol is 3 to 4. So if we double that, it's 6 to 8. So adding those together, right, we get 9 to 12. 9 to 12 is clearly less than 18. So this is clearly saying that this is going to be insoluble. In actuality, the solubility is 0.015 grams per liter. Remember we said, so that's 15 milligrams in a liter. Remember we said the level of solubility was 33 milligrams in a milliliter. So this is, what, 2,000 times less soluble than our cutoff for being soluble. So yes, it is very insoluble. Now, the numbers which are shown here, notice these are in ranges. This is already an approximate method. So this is my take on modifying this a little bit, is that I will give you the table here for the solubilizing potential for polyfunctional groups, but I'm giving to you them as a single number, which I think is a lot easier to work with than a range. Okay? I mean, you can see the differences between the monofunctional and polyfunctional, so the note here is that clearly monofunctional is not appropriate for this molecule. Okay. So this is my list, which is a modification of the lists from a table in the Lemke Functional Groups book, which is notated down here at the bottom. And so if we have an amine or some type of amine base that is protonated, it is a cation. So we're going to call it an iminium cation of some type. Now, this is going to include other types of things that are not specifically iminiums. In other words, if we have a pyridine and it's protonated and it becomes a pyridinium, we're going to count that as the same thing. Okay, I mean, I didn't make a complete list here because there's actually a wide variety of heterocyclic amines that, if protonated, would fall into this category. Basically, what we're talking about is cations, if we have a cation in our molecule. So focus on that. That will solubilize 25 carbons all on its own. That is a whopping amount. Now compare this to our unprotonated amine. So this is our typical amine down here in the middle. That by itself will only solubilize 3 carbons. So you can see there's about an 8-fold difference in solubility by basically putting a proton on this and introducing the charge. Big, big difference. Now carboxylate anions also increase the solubility by becoming an anion as opposed to the carboxylic acid, which is protonated. And if you see, the difference here is 3 to 12. So it's only a 4-fold increase in solubility. So you can see that the effect of an iminium cation is twice as much as we have for a carboxylate anion. Okay? So all that you have to do is, in your molecule, when you look at it, to find these groups, right, that are in there, add up how many there are, multiply it by the factor, and that then will give you the total number of carbons that can be solubilized, right? And going back to how do we determine, what are we talking about with carbons? What we're talking about is this number. It is, when we take our molecular formula, how many carbons are in that molecule, right? We have 6 in this ring, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, and up here 18. So there are 18 carbons in estriol. What this would also mean is, let's say we had no hydroxyls and we just had like a single amine group off of one of these, right? This was not here. This was just a complete hydrocarbon, right? And we put one amine group on here, doesn't matter, anywhere, and we had it acidic enough that it was protonated, that would be soluble because it would have a solubilization factor of 25, which is greater than 18. Okay? So that's how this whole system works, and that shows you also the power of these amines that are present in drugs, and many, many drugs have these types of weak base amines in them, or actually strong base amines, one or the other, that are important. A lot of natural products contain these types of amines in them. It's a really common component of compounds which are drugs. Now, not every drug, but I'm just saying it's there, and it's there often for a reason, because it gives us such good solubility characteristics. Now, the other aspect when we're talking about salts, right? So now we're talking about these ionized materials, right? So if we have an iminium salt, what kinds of salts, what could be the counterion? Well, it actually matters what the counterion is. If the counterion interacts again with the cation very strongly, then the material is not going to exhibit that strong of a solubilization factor. Now, we don't have solubilization factors for each of these different salts, right? So what it is, we're going to say the large solubilization factor that we talked about, right, is going to apply to all of these type of soluble salts. Succinate, tartarate, maleate, actually chlorine, acetate, and sulfate, that comprises probably most of the types of salts you'll see of drugs, right? These others do occur, not as frequently. If you just do a list, look at the list of drugs and the salt forms that they're in, you'll see the majority are in chlorate, acetate, or sulfate. Insoluble salts would be this pamoate or hydroxynaphthoate. So hydroxynaphthoate is very similar to this, it just has an extra hydroxy group on it, okay? And this is pamoate. So what these are is, even though these are combined, right, these are carboxylic acids, right, which would basically make a salt with the iminium group. But you can see these are big, these are greasy, right, and there's multiple forms, so they could actually form multiple bonds with compounds, making very large molecular weight complexes. These are insoluble. Okay, why would we want an insoluble salt? We might want an insoluble salt when we want something that dissolves slowly into the body. That just kind of serves as a depot. That might be a situation where we want that. Okay, so remember pamoate and hydroxynaphthoate as insoluble forms, right? The others pretty much are going to be soluble. Carboxylate salts, same kind of thing. Typically we see sodium, potassium, ammonium. These are all very soluble. So again, just more try to remember which ones are actually insoluble. So metal carboxylate salts can be relatively insoluble. Calcium, magnesium, these are the types of things that are less soluble. So that's the thing to remember there. And why do we consider this various salt forms? This table is from a reference, and we don't need to go through every aspect of it, but basically we can consider it for what is the oral dosage form we want. Do we want to make it an injectable? Do we want it to be in a suspension? You can see some of these other names here are for things that are for suspensions. We want it as suppositories. What is the dose range of the drug? Do we want rapid release or do we want slower release? So these are the relatively insoluble counterions, right? That's what we just said, like as a depot, they're slower release. We don't want to necessarily use sodium salts in hypertensive drugs, right? Because we're trying to get rid of sodium in the first place. So there's a lot of these types of considerations that we have that for our choice of salt. Now, this was just one of those tables with regard to what is the percentage of the anionic forms that we see in different drugs that are approved, right? So this is from USP. It's a while ago, but it's not going to be too much different nowadays. And you can see the majority, the largest percent is hydrochlorides, right? And so that's going to be the largest. We see acetates up here, right? And so on. So those are some of the biggest ones. Now, for cationic salt forms, again, typically, sometimes it's just as the acid form itself. In other words, it's not really a salt form. Or the sodium salt form. Potassium's also up there as well. A few cases are the calcium forms. And then, of course, this is miscellaneous, so that's why it's this large, because it's a big list, relatively speaking to everything else. So, again, either not as a salt or as a sodium salt. Now, when we combine various acids and bases to create salts, we might want to ask whether or not that salt, when added to water, might either change the pH or not. Now, if we have, remember we defined strong acids and strong base. These are materials that totally ionize when placed in water. So if we have an equal amount of a strong acid and a strong base, it makes a salt that also completely ionizes in water. So if we add it to water, neutral water, the pH will not change and it will be neutral. Okay? Because both are completely ionized. If we have a strong acid and a strong base, a strong acid and a weak base, the strong acid will completely ionize, but the weak base part of it will be partially ionized, which means the strong acid will predominate and it will act essentially as an acid and decrease the pH from our neutral point of 7. So atropine sulfate, atropine is a natural product. It's an alkaloid which has a weak base as a component of it, and sulfate is the salt form that it's often seen in. Now, if we have a salt of a weak acid and a strong base, now it's just the counter of what we had before. We have now essentially the base will predominate because it is completely ionized, and the acid is only going to be partially ionized. So in this case, sodium, like sodium hydroxide, right, is our strong base, fully ionized, and phenobarbital is a weak acid, will be partially ionized. So again, the pH will be greater than 7. It'll go up because the base predominates. Okay, so that's pretty straightforward. If they're equal, both strongs, it doesn't change. Stronger of the either acid or base is going to push it in the direction of either acid, lower than 7, or base, greater than 7. Now, the last case is basically very hard to determine. If we have a weak acid and a weak base, generally it's going to be very difficult to say exactly whether or not one or the other will predominate. You can make various types of arguments, but the point is that it's not as simple. So we're not going to make any kind of general assumption as to which is going to predominate here, right? Because there's different degrees to weak acids and weak bases. And so it's going to relate to which one is more ionized versus the other, but both are going to be partially. So we're not going to make a prediction. We're just simply going to say we're not going to make a prediction based on this type of a situation. So just remember the first 3 of these. Now, again, we're going back. Our definition, we have mentioned this before when we talked about functional groups. An acid, we're talking about a Bronsted acid, is a molecule capable of donating a proton. And a strong acid is one that completely dissociates in water. A weak acid, and there's different degrees of weak acid, is ionized to a level that's based on the pH of the solution versus relative to the equilibrium constant of the acid. This is a dissociation constant of the acid. Okay? So if the pH is less than the pKa, right, it'll be more protonated. If the pH is higher than the pKa, it'll mostly be deprotonated. So the amount that is protonated or deprotonated varies based on the relationship of the pH and the pKa. If we have a base, it's a molecule having an available lone pair of electrons capable of forming a covalent bond with a proton. So in other words, essentially taking a proton. And a neutral base, when it is protonated, becomes a conjugate acid. Okay, so these are Bronsted bases. A strong base will be completely protonated in water. Okay? So what does that mean? Hydroxide ion, right? So something like this is a very strong base. A weak base will be again protonated based on the level of the pH versus its pKa of that base. Other terms are amphipathic, and this is just a molecule with both hydrophilic and lipophilic groups. This is a very common feature we're going to find in folded proteins. A lot of times these amphipathic regions are what drives folding, with the lipophilic part going inside of, say, a globular protein and the polar parts on the outside. Or these could interact, say, with lipid, where, again, the lipophilic group interacts with the lipid and the hydrophilic part interacts with water again. It can be amphoteric, which we mean a compound that acts both as an acid and as a base, right? So it contains both of those types of functional groups. And a zwitterion, a neutral compound having a formal electrical charges of an opposite sign. And the simplest example of this would be a free amino acid. If we have a free amino acid, the amino group is protonated because of its pKa, and the carboxylate group is deprotonated based on its pKa. They both exist in one molecule. The net charge is 0, but each of them has a formal charge in each of those two atoms of the molecule. So that's a zwitterion. Log P and log D. So most of the time you'll see people talking about log P. You'll only see log D usually talked about in actual papers that are referring to the drug substance and various analytical analysis of the drug substance with regard to its solubility. Because in reality, when we do log Ps, we're determining them in a solution that has a particular pH. And if the compound has ionizable groups, then some of those groups may be partially ionized or fully ionized. Log P technically is only a measure of the octanol water concentration of non-ionized molecules. So in reality, for any compound that can be ionized, what we're actually measuring are log Ds, and we have to report what pH this was reported at. It'll often still just be reported casually as a log P. So we just be aware of that. So log P is a formal definition, but it is also casually used when talking about a log D at a particular pH. So just be aware of that possible confusion point. Okay, so log P, for example, measured log P, right? We are taking equal amount of octanol, equal amount of water, putting some fixed amount of drug in it. It will dissolve. Certain amount will dissolve in the octanol, certain amount will dissolve in the water at equilibrium, right? So practically speaking, what we do is we put all three of these together, we shake it up, mix it, and then allow the two layers to separate, and then actually determine the concentration of the drug in the two different layers. Then what we can do is look at what is if the amount of drug, right, relative to the amount of drug in water is at a ratio of 10,000 to 1, then P equals 10,000, right, which is 10 to the 4th. So log P will be 4. So if we see a log P of 4, what this means is we have 10,000 times as much drug in octanol as we do water, right? It's very much more in the octanol. A log P of 2, right, is 100 to 1. Log P of 0.5 is 3 to 1. And log P of 0 is 1 to 1. And we can have negative log Ps. In other words, if more of it is in the water than in the octanol, right, it could be, say, 1 to 10. If it's 1 to 10, then log P is going to equal -1. Because the 10 is going to be in the denominator, 1 is going to be in the numerator, and we take the log, it becomes the minus. So we have, when we're talking about passive absorption, in other words, no active transporter, not recognized by proteins, it's just going into lipid and then through the cell and then out the other side of the cell. There are log P ranges which are considered kind of optimal for that particular type of penetration. And the type of tissue that you're going across differs, because the permeability of different types of tissues and membranes is different, because the components in those membranes is different, as well as the thickness, a variety of other types of things. So CNS penetration, we're usually looking around, we want 2 or so. Oral, not much different. Intestinal, maybe a little bit lower, right? A lower number is more water soluble. Colonic, again, a little 1.3-ish. Sublingual, now this is where our lipophilic drugs get absorbed. And what types of things do we do sublingual? Well, melatonin tablets, nitroglycerin, things like that. So where there's a higher log P, this is actually one of the preferred methods of administering it, a high log P drug. Percutaneous, so this is across skin, 2.6. What do we have that goes across skin? Now, usually we're also adding some other types of chemicals to, say, a patch to get it to go across skin, because the skin is a very, very difficult tissue for things to get across. So they tend to be low molecular weight compounds, and log P around in this range is ideal. So one of the most common ones you might think about are nicotine patches, right? So nicotine is a small, essentially alkaloid that is used in this way, where for smoking cessation, nicotine patches can be given. Okay, consequences of dosing. So if our log P is less than 0, now notice it's lower than most of the optimal ranges that were mentioned previously. So very often these compounds will be seen as injectables. In this middle range of 0 to 3, right? So this is kind of in the spot where most of those compounds were, right? Typically those will be oral. If it's in the higher end of this is what we were talking about, transdermal, potentially. And if it's very high, we have to be concerned about toxic accumulation in fatty tissues. In other words, this is now so much more soluble in octanol than water, it doesn't really want to go in water. So where is it going to go? It's going to tend to stick in membranes, but also the other places in fat. And one thing you can often think about that would be like this would be essentially cannabis materials, right? So it's the thing you may have heard that basically if a person uses fair amounts of cannabis, right, it can be detected in their body even a month later after they've stopped taking it. That's because they've accumulated it in their fatty tissues, and then slowly over time is it only able to get out of that fatty tissue. It will, but it just constantly leaks out at a low rate, and so therefore it is detectable over that time. So that's just a demonstration that we do see accumulation like that for very high log P drugs. Okay, so now we're talking again about our log P versus log D. So what are we actually talking about here again? Remember we said our log P is, notice we say AH. So this is an acidic drug that's protonated. An acidic drug that's protonated. So it is not deprotonated at all, right? It is the concentration of the protonated form and neutral, and the protonated form neutral in octanol versus water. So it's the, right, this up here is all that we're seeing. So that's all that the log P is measuring. Now, in reality, this is an ionizable drug, and depending on the pH we have of our solution, right, a certain amount of it is going to be here. Now, not much is going to be here, because this ionized group is not going to be very soluble in octanol. So this is going to be a very small number, such that really it's these three components that drive what the log D would end up being. So what is the difference between log P and log D? It's going to be the difference that's created from the amount in water that's ionized. Right? And so you can see that's in the lower part. It's a little easier to see, right? It's the log P, but then we have a fudge factor, right, which is talking about 1 minus the fraction that is ionized. So in other words, it would be essentially the percentage of this form versus this form in the water. That's what we're talking about. And we can determine this fraction ionized based on 10 to the pH minus pKa. That'll give you an estimate of the amount ionized. Okay, so that's one way we can do the relationship. I wouldn't typically expect you to necessarily know this. This is not critical for most things that you need as a pharmacist. If you're determining a log D, what you're really doing is you are measuring actually, right, the concentration of this versus this at a particular pH. I mean, actually it's all of this, but like I'm saying, this is going to be much larger than this in the octanol. So when you're doing the log D, you're considering all of this, and it's going to be dependent on the pH that you have in your aqueous phase. And that's actually, you know, from an experimental standpoint, what you're measuring. So log D depends on pH if the compound is ionizable. Okay? And usually it will be less than the log P when the drug is ionized. Does that make sense? So from the standpoint, remember what happens when we ionize a drug. If we ionize a drug, right, it should be more soluble in water than this, right? When we talked about our solubilization potential, it was about this is 4 times more soluble. So what does that mean? That means that if we can ionize this compound compared to only having it in this form, right, more of it's going to be able to be in the water. That means the denominator of this equation for the log D is going to be bigger than the denominator here where it's not ionized at all, right? Because now this is more water soluble. That means if this denominator is smaller, this log factor becomes smaller than it would be here. So that's what we're saying. If we're ionizing our compound, then our log D is less than the log P. If our compound is not ionized, log D equals log P. And this is what it looks like as we titrate a compound. So here we have a compound theophylline. It's a pK of 8.8. And the log D here looks like the log P at pH values leading to the unionized drug. Okay, so pK of 8.8, that's right here. Okay, and this is now, if we go to the log D, right, the log D gets close to 0, which is actually what the log P would be as well. When we get up here and we start ionizing our theophylline, right, it becomes more charged. Now notice the log D goes down, right? This is going to actually mean there's more in the water than there is in the octanol at this point. Especially since these values are all actually definitely negative, right? At 0, they're equal. There's an equal amount in the water versus the octanol. And here, anything below that, there's more in the water than there is in the octanol. So this is showing exactly that when we protonate it, we're now at a situation where our log D equals our log P, and here our log D is less than the log P. And if we have a more complex compound, so this has a phenol, this has a carboxylic acid, and an amine, right? These are different pKa's, a pKa of 4, 9.44, 10.78. And as we titrate through there, you can see how this log D goes up and down and up and down. And all of this is based on the amount of ionization that we have in the molecule, right? And what is it going to be? Let's just take this all the way from the 0 state. In the 0 state, we're going to have all 3 of these protonated, right? So it's actually going to have one charged group here at 10.78. So it's going to have a +1 charge here. Then when we go, oh, there's another base here, 5.27. So actually it'll have 2 plus charges here. So your log D is actually a negative number. Now, as we go up through here, what's the first thing that's going to happen? Well, the first thing that we're going to hit is this 4.04, which means we're going to titrate this and make a minus charge. So our net charge is going to go to +2, and then we're going to add a minus charge. So it's only going to be +1. So it's going to be a +1 charge up here. And then as we go up here, we're now removing this proton, right? And we now have a +1 and a plus and a minus charge, right? So actually it's a net 0 charge. And then when we go up here, we're going to titrate this next one, which is the 9.44. So we're going to have another minus charge. And then when we finally get all the way up here, these two will be neutral and these two will be minus. And so this will be a -2 charge here. So this is a +2 charge, this is a -2 charge, and then every inflection point we have a change in the net charge of this whole molecule. And you can see how the log D of that molecule changes as we go through those different ionization states. So like we said, if we have a compound that's in water, it's very important that we state what the pH is, particularly if it's ionizable, because the log D will be dependent on that. So now, let's just go back, and like I said, most of the time you're going to see log P's, and this is what we're going to talk about. So just be aware that sometimes when we're talking about log P's, we're talking about log P's at a characteristic pH, which is actually a log D, but we're talking about here log P's. So these are just examples. This is not absolutely important that you would know any one of these particular examples. It's just showing you what does a compound look like in these different ranges of log P. Right? So highly water-soluble drugs where we have a very low log P, in this case, tobramycin here is -3.44. It's an intravenous drug, which is for cystic fibrosis patients. It's not orally absorbed because it is so polar, and you can see it's got all of these polar groups attached to it. Okay? Sorbitol, right? These are basically natural sugars that, again, these are all materials that would be very, very water soluble. -2 to 1 range, again, cyclovir, oxytetracycline, so on. This is just going to be intermediate, but in general, this is still quite a polar compound, right? We can see we have an amine group on an aromatic. We have a couple of carboxylic acids. So this is a reasonably polar compound and actually is more soluble in water than the octanol. The other thing is, even within classes of drugs, you can see ranges, and it's actually sometimes there's optimal activity based on an optimal log P. So this is a bunch of barbiturate drugs, and as the structures change, so does the log P, and also then the percent absorbed, right, is a function of this log P. So from this standpoint, you can see what's going to be ideal. The ones that are higher absorbed, right, turns out to be the ones that have a bit of a higher ratio in this case, chloroform instead of octanol, but it's the same idea of basically lipophilic versus polar solubilization. So the ideal here seems to be going up in this trend direction. As it turns out, as you modify the compounds and make them even more lipophilic, right, then what happens is this thing usually tops out somewhere and then turns over. So there is an optimal somewhere. This chart is just not showing that. This is just showing one side of this chart where we go from essentially compounds that are more water soluble to less soluble. So the specific ones that we're showing here with numbers, This is a log P of 1.71, and this is 2.33. So these are not super far apart, but you can see that as you go up, we're getting better absorption. Highly lipophilic drugs, this is the stuff we're talking about that can be potentially toxic. So you got to be careful. So generally therapeutic agents are less than 9, usually much less than that, usually less than 5. Amiodarone is an antiarrhythmic agent, slow and incomplete bioavailability. It's slow to reach its peak plasmic hours, right? And steady state levels may not be reached until 1.5 months of daily therapy, right? So what's happening during this? This is accumulating, right, in fatty tissues and things like that. So it has a very large volume of distribution. So you only have so many liters of blood. We're not 70 liters in the body. So what this means, not 70 liters per kilogram, right? So what this means is that this is accumulating in some tissue, and it's mathematically, when you calculate volume of distribution, right, it is basically looking as though the volume that it's occupying in the body is much, much larger than the volume that even exists in the body. So that's a hint as to accumulation in some tissue of the body. Again, this is basically another material that's very lipophilic. It's an antimalarial. Absorption is very poor, and basically if you want to get it in, you usually have it with a high-fat meal. This is true for a lot of very lipophilic drugs, and also some things like if you had it ideally with fatty vitamins like vitamin A and vitamin D, their absorption is better when you're having a meal with some fat on it, as opposed to having on an empty stomach with just water. And then our last topic is bioisosteres. These essentially are either atoms or functional groups having similar chemical or physical properties relative to another group in a molecule, resulting in them having similar biological properties. Now, this is going to be very dependent on what that functional group does in the drug molecule and how it's interacting with the receptor. In other words, where it's important. For example, a chloride ion attached to a molecule might be important just simply for its steric size, or it might be important for the fact that it's an electron withdrawing molecule and can lead to a dipole in a molecule. That can be different in two different molecules having a chlorine, right? So what might be suitable for a bioisostere might be different in two different similar looking compounds based on the fact that how that particular functional group is being used in the interaction with its receptor. So the purpose is to improve potency usually, or improve absorption or metabolic profile. In other words, this is not all of these. This is any one of these is sufficient. It could do more than one. Possibly to lessen toxicity, to simplify production or decrease costs, or to provide a patent position. In other words, somebody has patented a particular compound very well, and if you can put a bioisostere in one of the positions and they haven't covered that, you can potentially patent that bioisostere and then market it as a drug. You would presumably have to show some benefit for having put the bioisostere in, but still you could do that without violating the other person's patent. So 1 and 2 atom replacements. So if we have a methylene in a molecule, we could possibly replace that with an O, an NH, or a sulfur. Or the other way around, if we have, say, sulfur, so say in a methionine side chain, we could replace it with a CH2, and that's an amino acid called norleucine. So there actually are peptide analogs that contain methionine naturally. Methionine has a potential issue that it can be metabolized to a sulfoxide, and often those peptides then become inactive. So one way to avoid that is if we put a CH2 group in place of that sulfur, it now becomes just an alkane and it's more stable. It doesn't suffer from that potential oxidation and deactivation. We could have 2 atom replacements, right? OH versus NH2 or SH, this type of thing. And you can see how they might have similar properties, but there are differences, right? Because while the first two can be hydrogen bond donors and acceptors, the first one's neutral, the second one is basic, and the third one is slightly acidic, and cannot or doesn't really involve in hydrogen bonding. So there are similarities and differences between them, and it's where they're similar that you can substitute them. Where the feature is different, then you can't substitute them. Carbonyl groups, then we have variations on that, C double bond, heteroatom, other combinations. And then we have various types of either halogens or electron withdrawing groups, in other words a CF3 instead of a chloro. Because fluorine is very electronegative, so CF3 groups are very electron withdrawing, very much like our chloro and cyano and bromo, for example. Now, functional groups. Now this is a big one that's commonly used. Carboxylic acid. So carboxylic acid is pK about 4.5. Okay, and we have tetrazoles that also have a pK of about 4.5, but they're a little bit more lipophilic, right? So if you needed something that was potentially more lipophilic and it might give it better biopharmaceutical properties, replacing the carboxylic acid for the tetrazole could be favorable. Another one in this range is a hydroxamic acid, now versus a carboxylic acid. So for example, if you had a zinc metalloprotease, carboxylic acids are not the best chelators. So a hydroxamic acid could substitute potentially for it and be a much better chelator and give you better binding energy. Now there's downsides of that particular functional group from potential dissociation and toxicity, but the point is sometimes that feature can be used. Now notice if we have to mimic the pKa, it's not as favorable because it is basically a weaker acid. So the carboxylic acid and tetrazole mimics the one another based on their pKa and action as an acid. Now sulfonamide, not sulfonamide, sulfonimide. And why do we say imide? We're saying imide because we have basically electron withdrawing groups on both sides. These now have a pKa of about 5 and are important in what we call sulfonamide drugs, where we need this acidic position here. If we don't have this other acyl group on here and we just have a sulfonamide, this is not very acidic at all. Okay, so this is basically just showing an unusual type of carboxylic acid mimic. So we talked about some others in the tetrazole. So this is an actual example for a thromboxane A2 receptor antagonist, where they took this carboxylic acid, made a variety of modifications, and you can see how basically the activity of it went through as they made these modifications. And basically they ended up in the end with these materials down here at the bottom that are used to replace this carboxylic acid. Now notice what we have here. It's an enol. It's an enol that has an additional carbonyl group here. And another way that this could be drawn is you could have the ketone here, single bond here, and you would basically have an active methylene, right? So that's actually why this is very acidic and on the ballpark of this carboxylic acid, because this proton can get removed, and now instead of, it's really not an oxyanion as you would have if you had an alcohol, because actually through resonance this minus charge here can go down, make a double bond here, that double bond can come up and you get the minus charge here, and then this minus charge can go and make a double bond here and then push these electrons up, and then you have the minus charge here. So you actually end up with resonance forms of the minus charge distributed between here, here, and here, which then makes this a much more stable anion and similar to a carboxylic acid. So this is a very unusual and very interesting and well thought out bioisosteer in this case. Okay, here we have, say for example, an amide. So what are the features of an amide that we might want to mimic? Well, obviously the spatial characteristics, right, of where these two R groups are, but also that this is a partial double bond here and that this is planar. Okay, well, this one is going to be freely rotated. It's not going to be planar, even though it has similar spatial characteristics. Now this one will be, because this is a double bond and it's going to be fixed here. This now is replacing a carbon here with a nitrogen here, which may or may not be tolerated depending what you're substituting. And this is a thioamide. Or we have an ester if we don't actually need it to be planar, because this is now a freely rotating bond, and maybe we don't need the NH here, right? So it's possible that this could be substituted, this, or an ester could be substituted with an amide potentially. For aromatic groups, some common substitutions for them are, we have a phenyl and thiophenyl substitutes for that. Now this is a 5-membered ring, and you might think, oh, that looks pretty different, except for the fact that carbon-sulfur bonds are much longer than carbon-carbon. So in reality, in 3-dimensional space, if you looked at the CPK models, for example, right, those are those electronic surfaces, they look pretty similar between these two compounds. They're pretty overlapping. Now pyridyl is often substituted, sometimes in case of this, because now this is a weak base, can be partially ionized, can increase solubility, and aid essentially in absorption and the solubility. So that's a not too uncommon type of a modification that's made. So this is just one, this isn't a real example. This is just showing you if we had a compound like this and wanted to fully substitute out a bunch of the different groups for whatever reasons, we could say put a fluorine here in place of the hydrogen. That could lead to metabolic stability of not getting metabolism at this position. The double bond fixes this, and it goes from being just fixed to not being as polar as this. The thienol versus the aromatic, that's a pretty straightforward change. And instead of an ethyl, we have a methoxy, again, which may or may not be able to be tolerated depending on what's required here. But this is also pretty nonpolar. Even though it's hydrogen bond accepting, it's still an ether like this would be pretty acceptable in a lipophilic pocket. And then at last we have something which we call transition state mimics. If we have inhibitors of something, catalytic site, that we're often going to be talking about some stable compound that mimics or looks like the transition state. So if we have a proteolysis taking place, a transition state might look like this, where we have a tetrahedron here in place of the carbonyl of the carbon, right? This is the first step of hydrolyzing an amide. And so this goes into what's called an oxyanion hole, but this is not a stable material. This wants to fall apart. So a stable version of this would be something that might be, say, in this case it's a gem diol. Now normally this would be drawn often as an ether, but actually it exists as diol because of this strong electron withdrawing here, this trifluoromethyl. So these trifluoromethyl ketones, which is how they're sometimes often drawn, are actually trifluoromethyl diols. And so it's a transition state mimic. The other would be like, say, a phosphonate ester, right? So this is an ester, right? It's attached to another R group. It's in the chain, but it has this O minus here, which kind of looks like this O minus here. But this is a stable compound. That's the difference, and this is not.